Welcome to part 4 of our complete blood count CBC series. In this lesson, we'll focus on neutrophils. Neutrophils are granulocytic leukocytes and represent the largest population of circulating white blood cells. Because they are the first immune cells to arrive at sites of infection or tissue injury, neutrophils are often referred to as the first responders of the immune system. They belong to the innate immune system, providing a rapid, non-specific defense against invading microorganisms before the adaptive immune system becomes fully activated. Morphology of neutrophils. Under the microscope, mature neutrophils measure approximately 10 to 15 micrometers in diameter. Their most characteristic feature is a segmented nucleus, usually composed of 3 to 5 lobes connected by thin strands of chromatin. The cytoplasm contains numerous fine, pale pink to lilac granules that are not easily distinguished with routine staining. These granules contain powerful antimicrobial enzymes and proteins responsible for destroying microorganisms. Because their granules stain with both acidic and basic dyes, they are called neutrophils, meaning neutral staining cells. Neutrophil development, granulopoiesis. Neutrophils are produced continuously in the bone marrow through a process called granulopoiesis. During maturation, they pass through 6 developmental stages: myeloblast, promyelocyte, myelocyte, metamyelocyte, band neutrophil, segmented neutrophil. Under normal conditions, only mature segmented neutrophils and a small number of band neutrophils are released into the circulation. The entire maturation process takes approximately 6 to 10 days, after which mature neutrophils enter the bloodstream. Functions of neutrophils. Neutrophils are specialized phagocytic cells that rapidly eliminate invading microorganisms. When bacteria enter the body, damaged tissues release chemical signals called chemotactic factors. These signals attract neutrophils to the site of infection. The neutrophils first adhere to the blood vessel wall, then squeeze between endothelial cells in a process known as diapedesis, allowing them to leave the bloodstream and enter infected tissues. They then move toward the microorganisms by chemotaxis. Once they reach the bacteria, neutrophils surround and engulf them through phagocytosis, enclosing the microorganisms inside intracellular vesicles called phagosomes. The phagosomes fuse with cytoplasmic granules to form phagolysosomes, where powerful enzymes and reactive oxygen species destroy and digest the microorganisms. Neutrophils and pus formation. After destroying bacteria, many neutrophils also die. The accumulation of dead neutrophils, bacteria, tissue debris, and inflammatory fluid forms pus, a common feature of bacterial infections. The presence of pus indicates an active inflammatory response rather than the bacteria alone. Neutrophilia. An increase in circulating neutrophils is called neutrophilia. Common causes include acute bacterial infections, acute inflammation, tissue injury, burns, trauma, surgery, physical or emotional stress, corticosteroid therapy, and myeloproliferative disorders. Neutrophilia is frequently accompanied by an increase in immature band neutrophils during severe infections. Neutropenia. A decrease in circulating neutrophils is called neutropenia. Common causes include viral infections, chemotherapy, radiation therapy, bone marrow failure, and aplastic anemia. Left shift. During severe bacterial infections, the demand for neutrophils increases dramatically. To meet this demand, the bone marrow releases immature band neutrophils into the bloodstream before they have fully matured. This increase in immature neutrophils is known as a left shift. A left shift is an important laboratory finding that commonly indicates an acute bacterial infection, severe inflammation, or significant physiological stress. In healthy adults, neutrophils account for approximately 40 to 70% of the total white blood cell count. Neutrophils are among the most clinically significant white blood cells evaluated in the complete blood count with differential leukocyte count. Neutrophils can be measured using 2 methods: manual differential counting and automated analysis. Manual method. In the manual method, a thin blood smear is prepared on a glass slide and stained with a Romanowski stain, such as Wright or Wright-Giemsa stain. A medical laboratory technologist then examines the stained smear under a microscope using the oil immersion objective. The technologist identifies and counts 100 white blood cells, classifying each cell according to its type. The number of neutrophils counted is then expressed as a percentage of the total white blood cells examined. For example, if 60 out of 100 white blood cells are neutrophils, the neutrophil count is reported as 60%. Automated method. In modern laboratories, neutrophils are measured using an automated hematology analyzer. The analyzer examines thousands of white blood cells within seconds using advanced technologies such as electrical impedance, laser light scattering, flow cytometry, and cytochemical analysis, depending on the instrument. The analyzer accurately identifies neutrophils and reports both the percentage of neutrophils and the absolute neutrophil count, or ANC, which represents the actual number of neutrophils in a specific volume of blood. Thank you for watching. In the next lesson, we'll explore lymphocytes,